All right, so we're going to continue our talk on innate immune responses. So first up is inflammation. And the goal of inflammation is to essentially wall off or quarantine any pathogens that suddenly enter the body. Now, normally you're going to get inflammation if like the skin has been punctured, right? Kind of like that parenteral transmission. And so essentially what's going to happen is once the skin breaks, okay, certain immune cells are going to kind of sound an alarm, so to speak. And the swelling is going to help block the pathogen from spreading further beyond that initial infection point. So that's the goal of inflammation, to immobilize the bacteria or whatever the pathogens may be, until some of the phagocytes can get to the site of infection and clear those pathogens. So there are a few indications that inflammation is occurring or has occurred, and that would be the skin in that area is going to get red and warm, and then there's going to be swelling, which is actually going to be what causes the pain as well. And so you see there are some technical terms there associated with the informal definition, which you're going to want to be familiar with. But as we go through the mechanisms of what happens throughout the inflammation process, we'll talk about why these are the indications of inflammation, why the skin gets red and warm and all of that. All right, so when something punctures the skin, the presence of mast cells in the connective tissue is going to be what triggers an inflammatory response. Okay, so again, mast cells are found in connective tissue. So when that tissue breaks, the mast cells are going to release histamine into the area, and that is going to trigger vasodilation. So those blood vessels are going to dilate and become larger. This is going to allow a more rapid influx of white blood cells and oxygen and nutrients, right? Blood is going to be moving faster, hence the redness. But also, because the vessels are dilated, they're going to be leaky. And so some of the leaky fluid from these vasodilated vessels are going to move to surrounding tissue, which is what's causing the inflammation. But some of that is going to push on nerve endings, which is where the pain is going to come into play. So as you can see here, on looking at the right-hand picture, which is the increased permeability due to vasodilation, look at the cells that are lining the blood vessels. You see how there's little gaps present? Well, that is really important because with the increased movement of white blood cells, these little gaps are going to allow those phagocytic cells to be able to squeeze through the vessels and get to the site of infection, okay? So the actual movement of white blood cells through the vessels, kind of imagine them like marching down the vessel trying to get to the site of infection. That movement is called margination. You're going to want to remember that. And so the white blood cells are moving through the vessels, margination, and when they get to the site of infection, they squeeze through those gaps, and that squeezing through is called diapedesis. Okay, so those are two other inflammation terms you're going to want to be familiar with. So the margination coupled with diapedesis is going to allow the white blood cells to get to the site of infection and clear up those pathogens, as quickly as possible. So here's kind of a nice big summary picture of what is happening. So here you see the skin, right? And so the tissue has been damaged. Looks like by a rusty nail or something quite painful. But when this happens, mast cells are releasing histamine, okay? And so that is going to result in vasodilation, which is going to increase blood flow to the area. So what you see down on the bottom is you see the white blood cells that are moving along So that's margination. And you can also see them squeezing through, which is diapedesis. And then you can see that once they have squeezed through, they are phagocytes. So they're able to start ingesting and digesting the pathogen in that area. Okay. Now the phagocytes also engulf like dead cells, cellular debris, and things like that. Another important point of having these increased permeability or having an increased permeability of the vessels is because it allows fibrinogen to get to the wound. So fibrinogen is the primary clotting protein. So we want to cause a clot to again limit the spread of the pathogen. We're trying to keep it contained to one area. That way the phagocytes don't have to work too hard to hunt them down and clear the infection. So once all this has happened and the pathogen has been eliminated, then tissue repair can occur. And if it was like a small little, flesh wound, a lot of times, it can be completely repaired. If it was a deeper wound, a lot of times scar tissue will be present. All right. Next up is going to be pyrexia, which is the science term for fever. And pyrexia is a defense mechanism. So we usually say a fever is around 100.4 degrees Fahrenheit. And there are two really important key players with respect to pyrexia or fever. That's going to be the hypothalamus, which is the region of the brain that serves as the body's thermostat, and then little cellular messengers called pyrogens that are kind of like the, trying to be clever. Nothing is coming to me at the moment. They let the hypothalamus know that there's a problem and we need to reset the temperature, in this case, to heat it up. So there are different types of pyrogens. Exogenous is going to be pyrogens produced outside of the cell. So that means that the pathogen is, they're molecules made by the pathogen that alerts the body that there's a problem. So LPS, remember that endotoxin from gram-negative organisms, that's a very common exogenous pyrogen. And then there are also pyrogens that we make, and those are called endogenous pyrogens. So again, endo means within, so we make them within our own cells. Exo means outside of, so these are made outside of our body, so the exogenous pyrogens belong to the pathogen. All right, so let's talk about what happens here. Okay, so there's a little bit that I want to embellish on this slide that I'm going to want you to remember. So really the party gets started when an exogenous pyrogen is detected. So I would actually kind of pencil that in above number 1. The leukocytes, those are our white blood cells, our immune cells, and they have to first actually detect a problem. So when a leukocyte detects an exogenous pyrogen, then it's triggered to release interleukins. Interleukins are an endogenous pyrogen. We make those to let the body know, hey-o, there's a problem, we need to bump up the temperature. Okay, so let me kind of repeat that. So the leukocytes are the white blood cells that are kind of on the prowl looking for problems. When a leukocyte detects an exogenous pyrogen, like the LPS from a gram-negative pathogen, that leukocyte then releases interleukins, which are endogenous pyrogens that are going to circulate in the blood to get to the hypothalamus, the body's thermostat. This is going to result in the hypothalamus secreting prostaglandins, which are essentially going to trick the body into thinking, oh, it's too cold in here. We need to turn up the heat. Okay, so these prostaglandins are going to be what resets the hypothalamus. Okay, and so at that point, the hypothalamus is going to initiate those temperature raising responses. So what are those temperature raising responses? Well, the first is going to be peripheral vasoconstriction to the skin. And essentially what's happening there is they're trying to restrict blood flow, especially to our extremities, our fingers and our toes, because we're trying to minimize heat loss. Right, our extremities are normally a bit cooler because they lose heat a lot faster. And so we're trying to minimize that, okay? We're trying to increase our core temperature. So we're gonna do vasoconstriction to the skin, and then there's going to be inducing shivering, because remember, as you're shivering, things are moving very, very quickly. And so as molecules move, our temperature increases. Okay, so shivering is to raise our body temperature. And then the third thing that's going to be happening is there's a thyroid hormone that's going to be released from the thyroid gland to increase metabolic rates. Because remember, back when we talked about metabolism, I mentioned that all reactions as they occur naturally give off heat as just a byproduct. And so the more reactions that are happening inside of our body, the more heat is going to be released, and therefore trying to increase our core temperature. So what's interesting to me is actually what they call a fever breaking, right? Because at some point the fevers have done their job, right? And we don't need to maintain that temperature forever. So when a fever breaks, they actually call that the crisis phase. And I don't really know why, because it should be good that our fever is breaking. So I wonder why the person that coined this instance, why they called it a crisis phase. But essentially, when we hit the crisis phase and the fever breaks, we're trying to then cool down. So it goes more to vasodilation, right, to increase blood flow throughout the body. And we're going to start sweating, because sweating induces what's called evaporative cooling. And so sweating helps us cool down. All right, so why do we need a fever? Well, remember, the vast majority of human pathogens are mesophiles, okay? So when our body temperature gets to a certain point, some of those mesophiles can no longer grow, okay? So it's gonna inhibit the growth of some of them, but it's also gonna increase our heart rate. And so in doing so, right, the white blood cells and other important things are going to be delivered to infection sites more rapidly, like those phagocytic cells, for instance. Heat does increase the division of B cells and T cells. These are the key players of our acquired immune response. We'll talk more about them later, but it's kind of like we're kind of letting our acquired immunity know, hey, yeah, we're about to tap y'all in soon, all right? So we're kind of getting those cells ready. With heat, right, things are moving faster, so more reactions are happening, so enzymes can be more active. But also a fever makes you tired. And when you're tired, you're going to rest, because when you have an infection, you really need to be a couch potato. You need to let all of your ATP go towards our immune responses and fighting off this infection. The more you're up walking around and doing things or trying to be active, you're doing more harm than good. So letting a fever run its course and you being a couch potato and letting your body heal is going to normally reduce the length and severity of illnesses. Okay. All right, so you don't have to memorize all of this, but really this is just kind of to reiterate why you want to have and maintain a low-grade fever for a little while. Okay, of course, fevers need to be monitored. You don't want any crazy high temperatures for prolonged periods of time. But a low-grade fever is very, very beneficial because of all of the things that we just talked about, all right? So even though it's tempting, we don't like kids or significant others or ourselves to feel bad, right? You don't want to take a fever reducer at the first signs of infection, right? Especially because in younger kids, If they're not having a fever, they may not be as tired. So they're going to want to get up and move around and play, right? We all love to see kids and things happy. So you're like, yeah, cool, do it. I'm glad you're feeling better. But they're going to potentially overexert themselves, okay? And so when the fever reducer, has run its course, right, the pathogen, its numbers are going to be higher than previously because we're not fighting that infection as well. And so oftentimes the fever will spike higher than it was before because now there's more pathogen present, okay? So having a low-grade fever and letting it just run its course naturally is normally the best thing to do. But of course, monitor fevers. All right, next up, we're going to talk about NK cells, which are natural killer cells. And what's really important with natural killer cells is their ability to distinguish self from non-self. Okay, so we talked about this in an earlier video, but just want to kind of talk about it again. So a lot of self cells have a special molecule on their surface. MHC1, major histocompatibility complex. And you don't have to remember what MHC stands for, but all nucleated cells, which are us eukaryotic self cells, have MHC1 markers present on the surface. That is how our immune cells are able to identify a self cell, because pathogens aren't going to have MHC1 on their surface. Okay, they're going to have those PAMPs. We also call them antigens because they're foreign molecules that shouldn't be in our body. So when a natural killer cell, for instance, runs into a cell and it's kind of checking to make sure everything's okay, it's like, oh, you've got MHC1 on your surface. Awesome, good job. You're supposed to be here. Go about your merry way. But if it encounters a cell that doesn't have MHC1, it says, you're not supposed to be here, and it targets it for destruction via various ways. But NK cells can also tell infected self cells, and that's because, especially if a cell is cancerous or is infected with a virus, what tends to happen in those two situations is the number of MHC1 molecules are drastically reduced or altogether eliminated from the surface of the self cells. So that way, the immune cell can say, ooh, hey, you're infected. You're going to need to be destroyed to prevent that pathogen from being able to spread. Okay, oops, sorry, I jumped the gun there. All right, so the natural killer cell is a type of immune cell. And again, it's going to be assessing different cells that it encounters. Are you a self cell? Cool. Well, maybe cool. Are you a self cell? Yes. Are you healthy? Which means, do you have the MHC1 markers? If the answer to that is yes, then cool, go on your merry way. But if it says, oh, hey, you're a self cell, do you have MHC1? No. Uh-oh, you're infected and must be destroyed. Okay, so that's what we're looking at here. So it can target bacterial cells it can detect bacterial cells directly, but also cells infected by a virus and cancer cells. Okay, so look at the top part of this picture first. And so you'll see that the natural killer cell has two different molecules on its surface. It has an inhibitory receptor and an activating receptor. So if the natural killer cell comes upon a self cell that has the MHC1 molecules, that means it's healthy. And so the MHC1, the shape of it is going to be complementary to the inhibitory receptor on the natural killer cell, because we want to inhibit the natural killer cell from killing that healthy, normal self cell. Okay, because as the name implies, natural killer cells, these will kill cells that shouldn't be there. Okay, so we do not want to kill the healthy cells, so we are inhibiting it from doing its job. That's why it's called the inhibitory receptor. Okay, but now let's look at the bottom picture. So we have an infected cell. In this case, it's been infected by a virus. And so, as I mentioned earlier, when this happens, normally these cells don't have MHC1 on their surface anymore. Okay, so instead, what the natural killer cell recognizes is just a different type of receptor, and this particular type of receptor is going to activate the natural killer cell because it's not the MHC1 molecule, which means something is wrong. So when the natural killer cell comes into contact with this other molecule, we call it the activating molecule, the natural killer cell is going to target it for destruction. Okay, and so it causes that infected cell to induce apoptosis, and that means programmed cell death. So as you can see, that cell will die. And what actually happens is when activated, natural killer cells use two special proteins. One is called a perforin, P-E-R-F-O-R-I-N, and it is going to literally punch a hole in the infected cell, okay? And it punches that hole so that the next enzyme called a granzyme is going to be able to get through that punched hole, and it is a protease, so it's going to go and destroy a lot of the important proteins inside that cell, causing the cell to die, okay? So please do remember the two enzymes associated with natural killer cells. And so we're going to end this video looking at some pretty neat pictures. So this is a cancer cell surrounded by a lot of white natural killer cells. And then on the right, you can see the natural killer cells, like the one on the right, attacking an infected host cell and targeting it for destruction. All right, y'all, that's the end of this video. Let me know if you have any questions.